morning, friends. In the last class, we discussed about the ascent of sap, that is the upward conduction of water in plants. We discussed different theories, and the most acceptable theory was the cohesion tension theory, and the tension created by transpiration pull. It is a wonderful process where the water reaches the root from the surrounding soil. It moves upwards through the xylem vessels, reaches the bundles of leaves, and then to the mesophyll tissue and intercellular spaces in the spongy tissue, and water escapes from the space to the outer atmosphere through the opening called stomata. This process is known as stomatal transpiration, and the stomata plays a very important role in this process. The stomata, which is the opening of the leaf to the atmosphere, has very important physiological functions. It helps in the osmoregulation, it helps in the gaseous exchange of carbon dioxide and oxygen. And today we discuss the role of stomata in the transpiration. Transpiration is inevitable, and it was described as a necessary evil by Dickson and Joly, who proposed the theory of cohesion and tension. Because due to transpiration, more than 80% of absorbed water is lost from a normal mesophytic plant. If there is no special mechanisms to check transpiration, large amount of water is lost from the plant. But it has its own physiological functions. That's why it is called as the necessary evil. So, today we are going to see the mechanism of transpiration, different theories regarding the transpiration, and the importance of transpiration in the uptake and conduction of water. Here you can see the section of a leaf where the stomata is clearly shown. The uppermost epidermal layer, the palisade layer, spongy layer, intercellular spaces, and the substomatal chamber are very clear. And the open stomata is also visible. The water escape from the leaf. So transpiration can be defined as the loss of water in the form of water vapor. There are different types of transpiration. Most important one is the stomatal transpiration, but transpiration can occur through any open space. For example, lenticels, the cuticle. But the primary purpose of these openings was described as the gaseous exchange. Lenticels are for the gaseous exchange in the case of stem, and stomata are for the leaf. And in the case of roots, when the roots are underwater, in such halophytes we can see some pneumatophores and pneumatothores for the gaseous exchange. So for gaseous exchange, an opening is required. And the stomata is the most common opening in plants. And the loss of water also takes place through the stomata. How this takes place? How this closing and opening of stomata occurs? These are the things we are going to see in this class. Carbon dioxide enters into the photosynthesizing palisade layer through the stomata, and the oxygen is expelled out or move out through the. It indicates that the stomata should be open at the time of photosynthesis. And the transpiration is a phenomenon which occurs when stomata remains open. So our question is how the stomatal mechanism is related to photosynthesis as well as the transpiration process. What is the cause and what is the effect? Some say due to photosynthesis, some chemicals are synthesized inside the leaf tissue and that is responsible for the opening of the stomata. On the other hand, water is required for photosynthesis, so a large amount of water is utilized there, and we cannot explain photosynthesis without the uptake of water. So this is the dilemma in explaining photosynthetic process and stomatal mechanism. We have a wonderful paper regarding the relationship between stomatal mechanism, gaseous exchange, and photosynthesis. The physiological role of stomata and transpiration in photosynthesis. I'm adding that link of that paper to your Moodle class. You can read the article. So we will see how the transpiration process occurs. First of all, transpiration is a energy requiring process. To what extent energy is utilized, whether it is purely a vital process or only the closing of stomata requires energy, or the signaling system requires utilization of energy. All these are important, but it is a vital process. Somehow energy is utilized. We will discuss those things also here. So let us briefly describe the process of opening and closing of stomata. As we know, the stomata opens when the guard cells become turgid. When the water potential of the guard cells decrease, water from the surrounding cells and the surrounding space move into the guard cells and guard cells become turgid. When the guard cells become turgid, the stomata opens. When stomata opens, transpiration takes place. Whenever the water potential of the guard cells or approaches zero, the water moves out from the guard cells. As a result, the guard cells become flaccid. When the guard cells become flaccid, the stomata closes. This is the basic process. But what we have to discuss is what are the driving forces which changes the water potential of the cytoplasm inside the guard cells. Because stomata opens when it is required. We can see the stomata opening in the morning. In many plants, the stomata remains open throughout the daytime, and during night, stomata closes down. In some other plants, which are known as the CAM plants, because in those plants, there is an accumulation of crassulation acids. Crassulation acids are 4-carbon acids such as malic acid, oxaloacetic acid, etc. In such plants where accumulation of these acids takes place, the stomata opens during night and it remains closed during daytime. So, stomatal opening and closing is controlled very efficiently, and our point is how this takes place. Those plants having stomata which open during daytime are called as photoactive stomata, and those plants which open stomata during night and which is regulated by the pH of the cytoplasm are known as photoactive plants. So we can say the CAM plants are scotoactive and most of the other plants are autoactive. You can see the closed stomata and open stomata here. So let us see the different theories proposed by different scientists. The first theory was proposed by Selig in 1963, which is known as the glycolate theory. According to this theory, in the absence of carbon dioxide, in the early morning, when sunlight becomes available, photosynthesis starts, the carbon dioxide is used up inside the leaf. So there is a deficiency of carbon dioxide inside the leaf. The glycolate is synthesized from the Calvin cycle intermediates. This glycolate undergoes the glycine-serine pathway, which raises the osmotic pressure or cause a decrease in the water potential in the guard cells. And water enters into the guard cell when the water potential decreases. So here the basic idea is that in the absence of carbon dioxide, glycolate is synthesized. And this synthesis of glycolate in the absence of carbon dioxide and excess of oxygen is a part of the photorespiration that we will study later. But we can see here an illustration regarding that process, but it is considered as a destructive process rather than which can be encouraged on every morning. So here you can see the ribulose biphosphate, which is a Calvin cycle intermediate, is converted to phosphoglycerate, which is converted into glycolate. The glycolate comes out of the chloroplast and moves to the peroxisome, where the glycosylate is formed. From the glycosylate, glycine is synthesized inside the peroxisome. The glycine moves out of the peroxisome and enters into the mitochondria, where it gets converted into serine. Then serine comes back to the peroxisomes. So this is a destructive process where the Calvin cycle intermediates are used up, broken down into simple molecules. Anyway, due to the presence of this glycolate, the osmotic potential increases or the water potential decreases in the guard cells. Water enters into the guard cell and the guard cell becomes turgid. This is a mechanism explained by glycolate pathway. But it is clear that there are some objections to the glycolate pathway. As I mentioned, basically it is the process of photorespiration. It is not advisable to happen every morning. At the same time, this theory cannot explain the opening of stomata during night in CAM plants. Because during night, there is no chance for deficiency of carbon dioxide inside the stomata because there is no photosynthesis there. There is no increase in the oxygen level inside the cells. So this theory cannot explain the opening of stomata during. Similarly, the experimental studies measuring the glycolate in the guard cells is applicable only to the C3 plants. In the case of C4 plants, there is no presence of glycolate during any stage in photosynthesis. So this is not fully accepted. All the processes are taking place in the guard cell. Also, all the process can take place. All are possible pathways. But this may not be the basic cause for transpiration. The next theory is known as the classical theory or a starch-sugar conversion theory. This is considered as classical theory because this was the theory used to explain the process for a long period. It was proposed by Lloyd in 1908 and it is a simple logical explanation of how stomata opens. In the morning, when sunlight becomes available, the cells synthesize simple sugars. Simple sugars such as glucose, fructose 1,6 phosphates. So all these simple sugars are osmotically active. As we know, the guard cells contain chloroplast, but the surrounding cells of the guard cells are the epidermal cells, which are devoid of chloroplast. Although the spongy layer below the epidermal layer have chloroplast, epidermal cells are devoid of chloroplasts. So there is no photosynthesis in chloroplast, but the guard cells have chloroplast. They can undergo photosynthesis. As a result, simple sugars are produced inside the guard cells. Normally, the water potential of the guard cells decreases so that the water present in the surrounding epidermal cells move into the guard cells. The guard cells become turgid and the stomata opens and the gaseous exchange takes place. During night, when this synthesized glucose and fructose are converted into starch, starch is not at all osmotically active. It is not readily soluble in water. It is a stored carbohydrate. So when starch is formed from these simple sugars, the water potential of the cytoplasm in the guard cells increases or approaches zero. As a result, water moves out from the guard cells and the stomata closes. This is a very good explanation, a logical explanation for the closing and opening of stomata. And this theory was, as I told, it remained acceptable for a long period. And in 1926, another scientist, Sayer, connected it with the pH of the cytoplasm. According to him, the pH of the cytoplasm increases in the guard cells during daytime and decreases during night. And the crucial role is played by the pH. And the basic idea is that higher pH increase the turgidity of the guard cells and increased pH opened stomata. This was the concept. One of the major issues raised against the theory of starch sugar interconversion was that the simple sugars which is synthesized during photosynthesis is considered as the basic cause of turgidity in guard cells in this theory. But in the early morning, without opening of the stomata, without gaseous exchange, how photosynthesis can occur? This was the major question raised against the theory of Lloyd. So another scientist, Scant, in 1932, related it with the respiratory process. Because when there is no photosynthesis, the cells undergo respiration. This increased rate of respiration increased the level of carbon dioxide in the cells. The pH of the cytoplasm increases. This increased pH, as explained earlier, decrease the water potential. During night, the carbon dioxide released during respiration is not utilized for photosynthesis. As a result, carbon dioxide accumulates in the cytoplasm and it increases the carbonic acid so that the pH decreases. A decreasing pH increase the water potential so that exosmosis takes place. So the lower pH favors the closing of stomata and the increased pH favors the opening of the stomata. This was the explanation given by Steyer. It was in 1948 another scientist, Yin, who for the first time connected it with the enzyme phosphorylase. It was observed that the activity of phosphorylase increases during the stomatal opening. So it was in the 1948 a study in that line was proposed for the first time. Phosphorylase enzyme phosphorylates the glucose produced due to the breakdown of starch using inorganic phosphate. Starch is converted into glucose 1-phosphate due to the phosphorylation activity of that particular. And with the help of this theory, we can explain the opening and closing of stomata during night also, because when there is a phosphorylase enzyme and phosphorylates the glucose so that the pH of the guard cells increase during daytime. So according to them, phosphorylase enzyme and inorganic phosphate plays the most important role, and the role of light is not that much important as Lloyd mentioned. According to them, starch is converted into glucose 1-phosphate in higher pH, and low pH favors the conversion of glucose 1-phosphate into starch, which helps in the closing of stomata. So the interconversion of starch and glucose is dependent on the pH of the cytoplasm. So this is a shift in the concept where the pH plays the most important role in the closing and opening of the stomata. The accumulation of crassulation acids and their transportation into the vacuoles plays a key role in the opening of the stomata in such plants. So this was another explanation for the questions raised against the theory. So according to this theory, glucose 1-phosphate is soluble in water, so it decreases the water potential or increases the osmotic potential. Water enters into the guard cells in high pH. The stomata become turgid, the guard cells become turgid, and the stomata opens up. In lower pH, the glucose 1-phosphate is converted back to starch so that the turgidity of the guard cells is lost and the stomata become closed. An important modification of this theory was in 1964 when Stewart proposed that the glucose 1-phosphate cannot decrease the water potential or osmotic potential. He said that the glucose 1-phosphate has to be converted to glucose 6-phosphate to make it osmotically active. According to him, the glucose 1-phosphate is converted into glucose 6-phosphate, then it is again converted to glucose and inorganic phosphate. Both are soluble in water. As a result, the water potential of the cytoplasm in the guard cells decreases from 0. We can summarize the process as: the starch is converted to glucose 1-phosphate. The glucose 1-phosphate is converted to glucose 6-phosphate. Glucose 6-phosphate splits into glucose and inorganic phosphate. Both dissolve in water, increase the osmotic potential or decrease the water potential. So influx of water takes place into the guard cells. Guard cells become turgid and the stomata opens. During night, photosynthesis stops. As a result, the carbon dioxide is not utilized. So carbon dioxide accumulates in the surroundings of guard cells. So the accumulation of carbon dioxide decreases the pH below 7, so that in the acidic medium, the glucose combined with inorganic phosphate to form glucose 6-phosphate, then to glucose 1-phosphate, then starch. So this interconversion, the conversion of glucose into glucose 6-phosphate requires ATP. So there is a utilization of ATP in this process. So closing of stomata is an energy consuming process. So by this time we can trace the concept is changed very much, where rather than the conversion of glucose into starch and starch into glucose, the interconversion of glucose 6-phosphate and glucose is the most important point or step in the closing and opening of stomata. And the role of phosphorylase enzyme is very much accepted as a major player in the process. But there are some objections to this theory. The most important thing is that the concept that the photosynthesis starts before stomatal opening. Although it is explained by many, but there are some other mechanisms to explain this drawback. But the explanations given were giving more importance to the pH rather than the presence of light. The most important activity, according to this theory, is that the decrease in pH due to the accumulation of carbon dioxide, that is formation of carbonic acid. But it is observed that the carbon dioxide cannot cause such a drastic change in the pH. So this is one of the drawback against this theory. Similarly, it doesn't explain the role of or the effect of blue light in stomatal. Again, the starch sugar interconversion is very slow process and it cannot account for the rapid closing and opening of stomata. Similarly, the latest theories showed that the presence of glucose is a major player in the closing of stomata, but glucose is not detected in many plants during the opening of stomata. So all these are different objections raised against the theory of starch glucose interconversion. So the next theory, the modern theory, is an explanation of the process based on the ion exchange. Here the major role is played by potassium ions and the process of ion exchange. But the explanation is very simple, just like what we have seen earlier. This theory was proposed by Leavitt in 1974 and was elaborated by Bowling in 1976. I have added the original paper of Leavitt in your Moodle site. You can download it and read. It is one of the classical papers in plant physiology. According to this theory, the opening of stomata is an energy consuming process. During daytime, carbon dioxide level decreases inside the cell due to photosynthesis. Then starch is converted into glucose or undergo glycolysis. Other than glucose, it is converted into glucose and then undergo glycolysis. As a result, phosphoenol pyruvate is synthesized. This phosphoenol pyruvate diffuses into the stroma and it is converted into other organic acids. So the pyruvate is converted into oxaloacetic acid by reacting with HCO3. Then the oxaloacetic acid is converted further into malic acid. This malic acid dissociates into malate and H plus ions. So the most important theory, the most important point in this theory is the production of this excess H plus ions. H plus ions are produced due to the dissociation of malic acid into malate ion and H plus ion. This H plus ions can be utilized for the exchange with potassium ions. This exchange takes place. As a result, an influx of potassium takes place, potassium ions takes place, potassium comes inside and the potassium is osmotically active. Increasing potassium level decrease the water potential so that influx of water takes place, the guard cells become turgid. Plus cations are balanced by the malate ion, or it is an influx of chlorine ion also takes place, and this H plus K plus exchange is an active process. It requires the energy. During night, carbon dioxide accumulation takes place due to the stoppage of photosynthesis. When carbon dioxide accumulation takes place, the phytohormone abscisic acid stop the potassium exchange. This is the major step in the closing of stomata. Abscisic acid actually closes the potassium channels by depolarizing the membrane proteins. As you know, abscisic acid is a primary messenger. It does not enter into the cell, but it is received by the receptors on the surface of the guard cells. And there are some secondary messengers inside the guard cells such as inositol and calcium ions. The calcium channels of the vacuole opens up and calcium ions comes into the cytoplasm. So there is a plenty of positive ions in the cytoplasm. When calcium ions accumulate in the cytoplasm, automatically the potassium channels get closed. This is a mechanism proposed. So this is an illustration of the process. You can see the chlorine channels, potassium channels, calcium channels in the vacuole. All these are interconnected, and the abscisic acid plays a very important role in the closing of the stomata during night. Another important thing is the presence of certain specialized proteins called as aquaporins in the membrane of guard cells. The aquaporins are the intrinsic proteins present in the plasma membrane of all kinds of cells from bacteria to human beings, and they regulate the passage of many molecules including water, ions, etc. So aquaporins are present in the surface of guard cells also. The rapid closing and opening of stomata in response to drastic changes in the environmental stress can be explained on the basis of the activity of these aquaporins. So the conformation changes in the protein due to the phosphorylation and protonation of residues such as histidine or serine opens some channels for the movement of water. Similarly, the microenvironment created around the stomato is also important. It is found that the same genes are regulating the development of stomata as well as the trichomes on the surface of the leaves. So, the trichomes around the stomata or around the guard cells create a microclimate so that the evaporation of water is highly regulated by this microclimate. In the next class, we will see the genes involved in the differentiation of guard cells and the structural proteins present in the plasma membrane. Thank you.